Hello, my name is Evstathio Ilian. I am a postdoctoral fellow in the Volatolomics Research Laboratory at the Department of Chemistry of the University of Cyprus. In this presentation, I will give you a brief introduction on the research topics that we are involved in. But before we go into that, please allow me to start with an introduction to the lab's members. So the VRL was established in 2016 and is led by Assistant Professor Arabios Agabiou. Other members of the group include Kyriaki Kaigidi, a PhD candidate, and Fodini Papaioakim, an MSc candidate. So our lab primarily focuses on the identification and quantification of volatile organic compounds, known as VOCs, in 5 main areas. These include the human volatilome, atmospheric air monitoring, indoor air quality measurements, food and beverage industry, and agriculture. So the air we all breathe is primarily made up of nitrogen and oxygen, with carbon dioxide contributing 0.04%. VOCs are low molecular weight organic compounds that are found in minute quantities in the air. The origin of these chemicals can be human activity such as breathing and industrial applications, but also all biological systems, other animals and plants do produce VOCs. Although only found in small quantities in the nature, VOCs can have a significant effect on the environment through the formation of secondary aerosols through oxidation with sun. But why should someone study VOCs? So in the food and beverage industry, approximately 95% of all organic content is actually small molecules, many of which are volatile and hence classed as VOCs. Changes in the presence of these VOCs in food products can be used as an indicator of the origin, the type, and possible handling or mishandling of these food products. Also, our own body actually contains a biological VOC sensor, and this is our nose and brain, which are able to detect the presence of VOCs in aromas in food. So we are trying in the lab to emulate our own nose and breath using analytical equipment. Also, in human health monitoring, changes in the amount of specific VOCs in samples, such as breath, can be directly linked with diseases such as COVID-19 and cancer. We are able to detect these small molecules using very sophisticated analytical instruments called mass spectrometers. As mentioned earlier, VOCs are present at very low concentrations in the majority of the samples that we are looking at, and this is why there is a need for sample pre-concentration methods to be used, such as SPME. With using these methods, we can capture the VOCs in solvent material, which is based on polymers, but to release them and to analyze them, we have to heat this polymer material up to 250 degrees Celsius in a process known as thermal desorption. The release of these VOCs from the solvent material is then directed towards the mass spectrometer for analysis. A picture of our setup is shown below. It is only through the use of such sensitive and specific instrumentation that these VOCs can be detected and quantified. Our current areas of interest lie in human health monitoring, food and beverage aroma profile, and environmental and indoor air quality measurements. Some examples of work we have done in the past include analysis of VOCs in human breath, urine, and saliva for disease monitoring. In the food and beverage industry, we use the aroma profile of various products to identify the region of their origin. For example, we have found differences in the VOCs of carrots, wine, and olive oil that are indicative of the country and/or region of their origin. In the environmental and indoor air quality measurements area, we studied the chemical composition of exhaled cigarette and e-cigarette smoke, the emitted VOCs from building material in new office spaces, and the presence of pollutants from consumer products in hair salons. Another interesting area of our research actually includes atmospheric chemistry monitoring using drones, and this was a collaboration between the University of Cyprus and the Cyprus Institute. Our work is also funded by external funding agencies such as the European Union, and we currently have three ongoing projects. The first one lab is looking into using thermal desorption GCMS tools for the detection of COVID-19 in breath. The second one, into DBP, is involved in the use of analytical instruments for the determination of disinfection byproducts in drinking water. And the third one is a Marie Curie fellowship awarded to myself for the detection of autism spectrum disorders in breath of young children. With this, I would like to thank you for your attention.